first observation was at the 3rd of September at 1 p.m. The water level of the aquatic chamber was at 18.5 centimeters. The conductivity was at 116 to 117. The pH level was at 5.49, and the dissolved O2 was at 1.5. The chamber had clean water, the fish was healthy and was moving rather quickly, and the plant seemed healthy. In the terrestrial chamber, the soil was at 22.3 degrees Celsius, the air was at 23.2 degrees Celsius, the barometric pressure was at 29.31, the relative humidity was at 100%, the CO2 per ppm was 33300 to 33200, and the O2 per ppm was 117897. The terrestrial chamber, the snails had moved to the top of the lid, the plant was healthy and very green, the spider with the egg sac had moved into the decomp chamber, and the other spider was missing, and the worms were not visible as they were underneath the soil. In the decomposition chamber, the spider had had babies, the mealworms were not visible as they were underneath the plants, the fruits were rotting, the polys were not visible, and 8 baby spiders were not moving, and the adult spider was not moving either.
On September 9th at 12:52 p.m., the water level in the aquatic chamber was 17.5 centimeters. The conductivity was 133 microsiemens per centimeter, and the pH level was 5.52. The dissolved oxygen was 0.0 milligrams per liter, and the fish seemed healthy. It was moving at a good pace, and the plant was starting to droop and had white stuff around it and the water. In the terrestrial chamber, the air temperature was 22.1 degrees, and the soil temperature was 22.1 degrees Celsius as well. The barometric pressure was 29.37, and the relative humidity was 100%. The CO2 was 44829 ppm, and the oxygen was 157,755 ppm. The snails moved towards the soil, and the plant was healthy with only 1 leaf dead. In the terrestrial chamber, the air temperature was 22.1 degrees, and the soil temperature was 22.1 degrees Celsius. In the terrestrial chamber, the air temperature was 22.1 degrees, and the soil temperature was 22.1 degrees Celsius as well. The barometric pressure was 29.37.
So on September 14th, 2010, at 1:10 p.m., the aquatic chamber had a volume of 17.5 centimeters of water. The conductivity was 135 microsiemens per centimeter. The pH level was 5.78, and the dissolved oxygen was 0.0 milligrams per liter. The water was very clear, the fish was healthy and it was moving fast. The plant was covered with white stuff and was drooping a lot. The spider that was found there last time is dead and is still there. In the terrestrial chamber, the soil temperature was 21.9 degrees Celsius, the air temperature was 23.0 degrees Celsius, and the barometric pressure was 29.38. The relative humidity was 100%, the carbon dioxide was 96512 ppm, and the oxygen was 171487 ppm. One snail seemed to be dead, the other 2 were up on the walls, the plant was drying and turning black, the crickets and worms were not visible. In the decomposition chamber, the temperature was 22.6 degrees Celsius. The spiders and the babies that it had were not visible. The fruit is still rotting, but it's not black yet, and the roly-polies are on top of the leaves.
The materials we used for the aquatic chamber was 1 betta fish, 1 aquatic plant, about 20 centimeters of water, about 5 centimeters of gravel, and 1 rope that was 3 centimeters inside the water.
In the decomposition chamber, we used 10 pieces of fruit, 30 leaves, 10 chunks of potting soil, 15 mealworms, 2 worms, and 6 pillbugs.
In the terrestrial chamber, we used 10 centimeters of soil, 1 rosemary plant, 3 snails, 3 crickets, 2 spiders, 1 which was about 2 centimeters, the other was about 4 centimeters and had an egg sac. There were 3 separate egg sacs, and there were also 5 worms.
Our conclusion for the terrestrial chamber is that all of the animals inside of it survived, the snails and the worms, but the plant, from lack of sunlight, had withered and died.
In the aquatic chamber, the water was clear, the fish was healthy and mobile, and the plant started to droop and grow white stuff around it.
So the conclusions we drew in the decomposition chamber were that the fruit started to rot in the beginning and continued to rot throughout the whole experiment. The mealworms grew very large as they were feeding on the decomposed fruit. The leaves turned brown, and the spider that had the egg sac moved into the decomposition chamber and had babies in the decomposition chamber, and the babies started to grow in the first couple weeks. With this project, we saw that all parts of the ecosystem rely on each other to survive.